Hi, my name is Krishna Shinoy, and it's a real pleasure to be here with you, especially in this highly unusual time. And I'm so happy that we're able to spend some time thinking about some science and engineering together. So I'll be giving the outro talk for week 2, day 2 on linear dynamical systems applications, specifically motor control and brain-machine interfaces. So I'll talk about four key areas: intro, a new conceptual framework which calls for this dynamical system perspective, which involves linear dynamical systems, review some recent results in this area, as well as their applications to BMIs. So movement is critical to life, and understanding cortical control of arm movements is important for two reasons. On the left here is basic systems neuroscience and neuroengineering understanding. And we're reminded of how exquisite our motor systems are by this picture of Steph Curry making a free throw, where you really have to get exactly right the trajectory, speed of launch, the release of the ball, and everything for it to go through a hoop and get paid a lot of money to do so reliably. But how do you do that? How do populations of neurons in the brain control the arms to do so accurately and reliably? And on the right, we seldom really appreciate these functions until they're lost. And this picture of Christopher Reeve helps highlight a translational neuroscience and neuroengineering opportunity, which is that he was thrown from a horse and paralyzed from the neck down, very sadly, in the mid-90s. And from that day forward, couldn't walk or move his arms or even breathe unassisted, and would like to simply understand the neural activity that's still present in the brain to drive prosthetic devices to help people with a variety of neurological diseases and disorders. So there's both basic science and engineering and medically oriented translational work, importance of involving these dynamical systems. Now, how do we study this? Well, on the left, if you want to reach out and pick up an object, a ball, for example, we see that object, that information comes in through the occipital lobe, gets elaborated into initial plans to move the arm in the frontal lobe, the premotor cortex, PMD, the dorsal aspect that I'll be telling you more about, as well as the primary motor cortex, M1. And then those plans are elaborated into control signals that are sent down the spinal cord to contract the muscles and move the arm. And there's a closed feedback loop. And we can tap into this neural activity in the brain using what is our workhorse, which are 100 electrode permanently implanted so-called Utah arrays. We can also measure activity with single electrodes or linear probes. And most recently, we've been using so-called neuropixel probes, which have over about 1,000 electrodes, and we can record from about 384 at one time, where the electrodes are so closely spaced we can do very accurate spike sorting. Okay, this is all in monkeys. We use monkeys because we can train them to perform elaborate tasks, and their brains are quite like humans. And we, of course, always have our eye on translating this to humans. Now, I'll introduce just one behavioral paradigm. This is all we need to know for today's talk. And this is called the delayed reach behavioral task, where a monkey looks and touches points of light on a screen in front of him, a so-called frontal parallel plane. After some period of time, a target turns on at one of a whole variety of locations. For example, one of 8 locations around a ring, but it could really be anywhere on the screen and so forth. Then there's some delay period, during which time he prepares to move his arm, but he can't yet move his arm until the central lights are extinguished and the peripheral target is made slightly larger or stops jittering. That's his go cue. And then from the time of the go cue until the time he starts to move his arm, not acquires it, but starts to move his arm, is called the reaction time, typically 250 to 300 milliseconds. And then he touches the target to get a liquid reward. Okay? And monkeys will do these tasks thousands of times a day. We can record from hundreds of neurons at a time, and their eyes and their hands. And it's a powerful paradigm. And you can do a lot of the same things in people now with clinical trials. So what is this new conceptual framework that I'm talking about? Well, let's review that in the visual system, if we shine a spot or a bar of light at different orientations, we will elicit in one neuron either a lot of action potential, shown as these vertical deflections, or none. And it's systematically organized such that if I plot that angle of the stimulus bar, I will map out an increase and a decrease again in firing rate, a so-called directional tuning curve, and there's a peak called the preferred direction. And this makes sense because the role of the visual system is thought to presumably be to internally represent the outside world. And neurons extract key visual information or features summarized by largely static tuning curves, meaning that they respond the same, independent of the contrast and so forth. Not quite true, but close to it. And therefore, the preferred directions are largely invariant to those factors, like contrast and speed of the moving edges. Now, that's the visual system where information is brought into the system. Now, it's very tempting to look at the problem the same way for motor acts, but very importantly, the causality arrow flows the reverse direction. Now the causality arrow is that neural activity causes the arm movement. So you can have a monkey reach to the left and to the right, and you can see that when this one neuron observation is measured during a leftward arm movement, you may get a lot of black ticks, a lot of action potentials being fired. Whereas to the right, you may find rather few, and there's 5 trials shown in each one of these. You can see the systematic arrangement around the clock. You can also map out a preferred direction. You could say, there is the preferred direction. This is how neurons respond. And Apostolos Georgopoulos, Andy Schwartz, many others absolutely move the field forward by showing that this is largely the case. But it's like a first order approximation. Where does it start to break down? Well, the role of the motor system presumably is to generate time-moving signals, or time-varying signals, like I said. And therefore, its job is not really to represent external information nicely and neatly for the experimentalists to analyze in individual neurons. Neurons generate or cause this time-varying signal, and most are not well captured by static tuning curves with simple functions. For example, the preferred directions, this peak here is not invariant in time to movement speed or to movement extent. And the tuning curves are therefore time-varying, complex, and heterogeneous, as many in the field have shown. So in other words, if I just make a leftward arm movement twice as fast, this preferred direction may change around a lot. So it's a little hard to understand how representation or tuning curves capture that. Now, John Cunningham and Byron Yu, 2 wonderful students from our group from years ago, wrote a beautiful piece in 14, and let me just read it to you. Population analyses are necessary in settings in which there may be neural mechanisms that involve coordination of responses across neurons. These mechanisms exist only at the level of the population and not at the level of single neurons, such that single neuron responses can appear hopelessly confusing, or worse, can mislead the search for the true biological mechanism. So what's being said here? It's not saying that what an individual neuron is doing is unimportant. It's that to understand what one neuron is doing, like one piece of a jigsaw puzzle, you really have to look at a whole ensemble or population of neurons to get the big picture. So that's the approach we've been pursuing for the last 15 to 20 years. And a dynamical systems conceptual framework, has absolutely been used before. It's widely used in engineering and physical science, and real early pioneers at Betz, Gilles Laurent, Bill Crestan have used these methods very productively in the past. And motor cortex, we felt, might constitute a dynamical system that prepares and controls movement, and in its simplest deterministic form, this can be expressed as some firing rate vector. So if you had 100 neurons under observation, imagine a column vector, one column wide with 100 rows, where in each element of it, you have the instantaneous firing rate of the neuron in some brief, at some time t. Now in discrete time, you could imagine that corresponding to a 10 millisecond time slice, and you're counting the number of spikes in that 10 milliseconds and then multiplying it to get spikes per second. So this is the response of the whole population of neurons. And it's saying that the time rate of change of that, or what in discrete time would be the firing rate at the next point in time, is some function, non-committal, some function, nonlinear, linear, we don't know, of that firing rate, plus some external input vector. Now, if we linearize it, we can write F simply as a matrix, and this is called the dynamics matrix. And if you recall, this was the focus of tutorial 1. It's simply saying the time rate of change of some neural population state, RFT, is equal to that firing rate state multiplied through by some matrix M. And we can study that M. So all of a sudden, our study of the motor system turns into fitting, learning, modeling what M is, and then investigating it for its eigenvectors, its eigenvalues, the forms it takes, and so forth. So the firing rates of each neuron depend on the firing rates of all other neurons. You can see that because a neuron number 13 here may depend on neuron number 22 here, and so forth, full mixing. So it definitely says that neurons depend on the firing rates of other neurons. It captures the time varying neural patterns, the temporal neural patterns, and how they might arise. Some neurons reflect the underlying latent dynamics. So it's saying that not all neurons have to be related to the external movement of the arm quite so neatly. They have to serve other functional roles of supporting the overall evolution. And some neurons may, but need not, display static tuning curves and representations. So it's saying that out of all these neurons, yeah, maybe some do look particularly neat and clean. That's wonderful. Okay, but it's not that it has to. So the prediction is that the neural population state should really reveal dynamical features. So the last setup slide here is to show you how it works. Imagine you had 2 electrode arrays, so now you have 200 rows, and this is one single trial, just over a second long. Here's 200 milliseconds. We track the eye, the arm, okay? And we can then go look in a 10 millisecond time bin, count the number of spikes in neuron 1, neuron 2, neuron 3 to get R vector, okay? And then we can slide that forward. And as we do so, we get a whole bunch of data. We get R's of T's at a whole variety of times. We get the time rate of change, because we can just calculate that from the data. And we can use that, okay, to solve for M. So if you recall, solving for M was the focus of tutorial 4. Now we can also, in addition to system identification or solving for M, we can also visualize it in some lower dimensional space. Now, whenever we take neural data at, say, 100 dimensions, maybe we could reduce it down to 20 dimensions without the loss of capturing much data variance. Okay, this is a fundamental concept of principal components analysis and other machine learning dimensionality reduction techniques. But if you keep looking at dimensions below 20 or 10 or whatever is capturing the vast lion's share of the data, you will definitely lose variance. So it's an incomplete picture. So when I show you a picture here in 2 dimensions, I am not saying that this is data that only lives in 2 dimensions. It's just saying I have to draw something, and these are the 2 dimensions that capture the most variance. Now, if you do that, you can start making statements like, okay, at the beginning of each of 15 different trials, I start at some point in red. I then evolve according to the gray trace, and I come out over here, okay? And over here, you could imagine there being an attractor state, which is holding at this location before a rightward arm movement. They would evolve out over here to the left, for example, before a leftward arm movement, just to keep the geometry simple. But they're different locations for planning. And then they arc around until the movement starts. And remember, neural activity leads the actual movement by about 150 milliseconds. So that's what happens when you are preparing and generating an arm movement. This dimensionality reduction was the topic of week 1, day 5. And then we also note that we could also reduce the dimensionality, maybe not to 2, but to 20, and then use those states to learn the dynamical system up here. And that can have some beautiful noise, denoising properties to it. Okay, so what are some recent experiments? First of all, do single trial neuron population trajectories progress lawfully? So here are these 2 dimensions, 15, I think, initial start points here, okay? And if I hit the go cue here, what I'm doing is I'm playing all 15 of those trials back at the same time just to show you the population average, okay? But they actually happen sequentially. They start here, they come over here, and I pause them and color them green where they're preparing, and then they cruise over here, and I stop them when the movement onset, and 14 out of 15 did exactly that. Now, one trial looked like the animal was planning to make that arm movement, for example, its rightward arm movement just fine, but then got distracted and did this loop-de-loop before coming over here. And if that's true, then we should find a much larger reaction time between go cue and movement onset, and that is indeed what we find. And what I'm showing here on the left, of course, is I'll always want to highlight key authors. This is Mark Churchwin and Byron Yu, and I'll put up some pictures as we go through, and then the final slide will be the real acknowledgement slide of all the real people that did the work. Now I can plot that over here at one time so you can see it. And again, you've already seen this plot on the previous slide, including this one aberrant trial that I've colored in red. And again, if you go analyze the dynamics, you might find that there's a fixed point attractor sitting right here that's drawing neural activity to it. But to come out over here, maybe there's something a little bit more rotatory that's going on. Okay. Second, do single trial neural population trajectories predict movement reaction time? Yes, they do. And how can we see this? Well, we have the hypothesis that if you, on a given trial, have to be preparing here, but you know you're going to head out in this direction, then if you happened by noise fluctuation or otherwise to be over here farther, you should have a shorter reaction time because you have less distance to go. And if there's some maximum speed, like a speed limit, that should be a shorter reaction time versus a trial that starts off farther away. That should have a longer reaction time. Moreover, if you happen to be here and moving in this direction, like you have a little momentum towards the starting line as a runner or something, you should have an even shorter reaction time than if you're in the same location going the opposite. So if you take that all into account, it turns out that we can explain about 4 times more reaction time variance than previous population methods. So this is setting behavioral data aside, making these neural plots, and then using the neural plots to go make predictions and test it in the behavioral data, and this is what holds up. Third, how is preparatory neural state arranged to initialize movement dynamics? So again, in this trial where he is planning and then moving down, I've shown you that data. But what about more real world conditions? Like what if he had to reach down on this curvature and then up to hit the target? Well, we created a so-called maze task where we could on the screen define a magenta maze, and he knew that if he moved his arm, which we rendered on the screen as a cursor, if he hit the magenta, he would abort the trial. So over time, he learned to move around it. And we also used colors to ask him to move fast or slow. That's what red and blue correspond to respectively. Now, a target could be in any location. We could use a whole variety of different complexities to the maze. And therefore, what we can get is we can get 108 different average trajectories going to different targets at different distances, different curvatures at different speeds. So it's basically like a white noise stimulus used in visual neuroscience, but it's a huge range of movements for motor neuroscience. And we'd like to see if theories hold up across all those, as opposed to being idiosyncratic to simpler types of movement. Now, when we go plot that in terms of these circles, which is where are you when you're preparing to make each one of those 108 movements on average, we find that they fall along a line. And if we say, okay, now start the movement, we see that no matter where you start, you rotate largely, okay, along a counterclockwise circle. Now, counterclockwise has no meaning because you could look at it in the other direction and see clockwise, but the point is that it's rotatory and it's consistent with there being one single dynamical system. And this is held up under modeling work that David Cicillo has done in our group with RNNs and so forth, and it seems to be a general motif. And we can take the neural data, we can go ahead and determine what M is, okay? And in this case, if we want to actually just restrict ourselves to the rotatory component, we can force M to be skew symmetric or just rotatory. And we find that this linear equation explains a large amount of variance, about 2/3 of the data variance, in the top two so-called JPCA planes. J means for rotatory. So this is one plane, JPC 1 versus 2. So this one plus the next one, JPC 2 and 3, captures about 2/3 of the variance, okay? So it's largely rotatory, and that's true even if we don't restrict the M. Now, can we do better? This is just the average. There may be 20 trials that goes into each one of these curves. Well, until recently, we couldn't do that, but we can now. It's called latent factor analysis via dynamical systems, or LFADS, and this enables inference of single trial neural population dynamics using sequential autoencoders and RNN methods to model the dynamics. And this is work done by Chetan and Pandranath and by David Cicillo. And what you can see here looks very much like this, except now you have hundreds and hundreds of individual trials. So happily, if you look at one of these average curves, it is representative of what's happening on all the individual trials. It's not like all the individual trials are all super noisy. We can actually peer back through that noise, and because we have enough neurons under observation, we can denoise it enough that we can actually see single trials. And this is a new powerful technique for moving this forward. Okay, now as a quick aside, in humans, it's possible to understand a little bit at least, and we think a lot, about speaking. So we wondered if we had a person say a whole variety of words, and we've published this recently, and we looked at where you were preparing before you said the word bat, or before you said the word bought, before you said the word beat, versus dot, or seal, or shot, do you see some orderliness? And in fact, you do. And lo and behold, again, you see this rotatory component. So it could be that this is a motif, okay? Because here we see that the neural state in 250 milliseconds around voice onset, so you're controlling the articulatory muscles in your throat and your face as you breathe to create voice. That's a motor act in recording from people during human clinical trials to help people with paralysis, okay? And we find that there's the same organization in speech as there is in arm movements. Now, fourth, can PMD and M1 firing rates change to prepare movement without affecting motor output? Okay, and this gives rise to these two concepts. If you read the technical note, you'll see what I'm saying, but what I'm really talking about are the null spaces and the potent spaces of the M matrix, okay, or here written as the W matrix. And this is exactly what's meant in linear systems or in linear algebra. And let's consider a muscle with two neurons. And if both of those neurons activate the muscle, then if I take the activity of neuron 1 up a little bit and number 2 down by the same amount, I don't change the net drive on that muscle, and therefore I don't expect to see any change in that muscle, and therefore it's so-called muscle null. It doesn't do anything to that muscle. And that line is the blue line, right? So if neuron 1 goes up, neuron 2 goes down, and that's this unit diagonal here. Now, orthogonal to that is the motor or movement output potent dimension, because if I increase both of these neurons' activity, I do increase the EMG activity and contraction force and therefore movement of that muscle and limb, and that's here. And so if we go look at our neural data, we see that at baseline we're sitting here. That doesn't tell us too much, but if we look at when you're preparing all those 108 different conditions, they all seem to line up along a line, along the output null line. And then when we say, okay, please move, we in fact move orthogonally off of that, okay? And Gamal Al-Said and John Cunningham have done some very nice work to show that this is actually orthogonal, not just different, but as expected from linear algebra. Okay, final part of the talk in the remaining few minutes is applications to brain-machine interfaces to help people. Now, millions of people worldwide face severe sensory motor impairments due to paralysis. And the causes of paralysis in the United States are as follows: stroke, spinal cord injury, traumatic brain injury, and so forth, and also included Lou Gehrig's disease, or ALS. Okay, so we'd like to help people. And the brain-machine interface concept is that if we know where in the brain, for example, premotor and motor cortex, as I've been showing you, individual neurons are encoding information about how you would normally move the arm, for example, to pick up that cup, but an injury, of course, keeps that activity from hitting the arm. Well, if we could read that activity out, if we could come up with a decoding algorithm and run it on low-power electronics that we could also implant, then we could use it to do a few things. We could move a paralyzed arm by stimulating the paralyzed musculature. We could move a prosthetic arm or a robotic arm, or we could move a computer cursor on a screen. Okay, so moving a paralyzed arm has been shown recently by Bolu Ajiboye and Frank Willett's wonderful work from Cleveland, where they read out the neural activity, translated it into stimulation patterns for electrodes on the arm, and the person was able to move his arm to pick up a coffee cup. Now, Lee Hochberg and John Donahue at Brown and MGH, and Jen Collinger and Andy Schwartz at Pittsburgh reported a number of years ago the ability to use a robotic arm or prosthetic arm, again, to drink a cup of cold coffee. I guess everybody loves coffee. Okay, so that's acting with the physical world, and that's very, very important, okay? That's very important. But what we've been focusing on is more the virtual world, where what we want to do is recognize the fact that if you actually ask yourself what you do these days, and have been doing over the last 10 years exponentially more and more of, is you interface with devices, phones and tablets and computers. So the ability to move a computer cursor or communicate really allows you to participate in an independent way in work, which is a real desire of people. You might not think that, but it is, because it's a sense of independence. Communicate with their loved ones and their physicians and so forth. So what I'm going to show you here is just a couple of samples of the work we have done with monkeys and a couple of samples of what we've done with people that all make use of the linear dynamical systems that you've been learning about, okay? So what you're seeing here is a target will come up on a screen that the monkey is sitting in front of. The monkey's not moving his arms, and he's going to be moving this cursor, okay? And he's going to move the cursor up and hold on that target to get his reward. And then the cursor will come to some other location. He'll do that, okay? Now, if we do that, I'll let it play. What you can see is that the cursor definitely moves out and hits the target. The target turns blue when he's in contact with it. Sometimes he flies right through it. But the cursor isn't that fast, and it's not that straight, and it doesn't crisply stop on the target. Now, what was the algorithm? Well, this was a standard velocity Kalman filter. A Kalman filter looks very familiar to you now. It's saying that some state, x, at the next time step is equal to the previous time step times some dynamics matrix, A, like the M was before. Now, if you want to look at the neural activity, it's related by some C matrix to the state matrix, X of K, where the kinematics are. For example, the velocity of X and velocity of Y. This is called the state update process, and it's called the observation process. Now, there's a noise, so we can add this W and Q, which are random and additive. And that's the standard Kalman filter, which is a dramatic advance about 10 years ago when it was first introduced, above and beyond other things. But it's still not that good. So what we did was create a system that can perform, as you can see here. And what you can see, and I'll tell you how it works in a second. And what you see is that the cursor is able to go straight, much more straight to the target, more quickly to the target, and is able to stop on the target and select it, such that in the same length of time, the same roughly 21 seconds, you're actually able to do twice as many selections. Or if this was a keyboard you're using, you'd be able to type twice as many letters, for example. And the whole key here was to still use the common filter, but to actually train it slightly differently. What we did was we started with the velocity common filter, but said, hey, those inaccuracies are probably because we didn't estimate all those parameters very well. So we ran it, just like you saw in this left movie, and you get this red trajectory here. But from the monkey's perspective, at every moment of the time, he's trying to go to the target, because it's the only target on the screen, and that's what he's trained to do. So we could reinterpret the neural activity that we got that actually drove this movement instead as it should have corresponded to moving directly to the target, these blue arrows. So we retrained or refit the common filter, and then didn't use that information online when we went back and ran the next experiment, as it was shown over here on the right. The other twist that we did, and this is not as important as the first one, but it's also a contribution to the performance, is that we did not propagate the uncertainty in the position, because vision is very good. You know where that cursor is, so there's no uncertainty, unlike velocity or other things, okay? So I'll just play it again real quick. Here you can again see that you are, yes, now going straight to the target, but it's not constrained to. It can go in any direction. It can go in curves and everything. It's, again, how we fit all the parameters of the common filter. It's not that we constrained it to only move along straight lines or something like that. Okay, now what about people? I have skipped over about 15 years of monkey-based work to get everything to work as well as possible, including high-speed target selection and so forth, with monkeys before we felt ready to go to people. But then we took all those advances as we brought them to people. And what's shown here is typing using this intra-cortical BMI. So the same Utah electrode array is implanted in this participant, T5, who is a wonderful early 50s Lou Gehrig's disease, person with Lou Gehrig's disease. And just during the experiment, we plug in this preamplifier, and these electrodes go to computers that I'm not showing you here, and they are moving this white cursor to the left or right or up or down or any direction here. When she's thinking about moving her right hand, which you can't do because she's paralyzed, to the left or to the right. Now, how do you click? The way you click on a target, like if I'm on the T, I want to select T, is she imagines clenching her left hand, which is just a strong signal that we found. And so we're moving that cursor using the refit Kalman filter, and clenching the left hand, we used a hidden Markov model, which I think you'll also be learning about, as the click signal. And what we were able to achieve is 24 correct characters per minute. So we asked her, how did you encourage your sons to practice music? And she's just thinking of her response and then typing her response. So when they started there, space. And what I'm showing you here is that this is when she's going at about 35 or so correct characters per minute. Note she has a delete key, so if she makes an error, she'll go back and hit delete to erase it. Now that performance fluctuates up and down some, and we're working to remove that. And there's a whole variety of challenges still left with these systems before they're ready for clinical prime time. But this is where it's at. This is the wonderful work of Chetan Pandranath and Paul Nuyujukian. Okay, and let me zoom in now to just simply highlight the keyboard that we used. And the keyboard that we use is the so-called Opti2 keyboard, which is an optimized human-computer interface keyboard. Okay, and what it does is it puts towards the center the letters that are more commonly used, keeping the Z and the X and the Q more to the periphery. And now we also did a copy typing test where we just said, when the sunlight strikes raindrops in the air, please just type that again. And what that does is that sorry, there's a hiccup with the movie. What that does is that removes her need to think of what to say, and we get 32 correct characters per minute instead of 24. Okay, now importantly, had we used a QWERTY keyboard like on your laptops, performance is a little bit lower because it's not optimized, but it doesn't fall apart, okay? And that's all been reported out. Now the final video, and then I'll have a wrap-up slide and we're done, is what we'd like to do is have a person be able to use any interface without being a custom interface. So with a Nexus 9 tablet running Android 5, okay, and a Chrome browser, and now for the first time, because browsers allow for word completion and prediction, we allow that to happen too. Before, we were only using raw information throughput because as, you know, not only good scientists, that's what we wanted to measure. not all the extra natural language processing that one can always add to it, which we certainly do. Okay. So she is not only an avid musician, but also an avid gardener. She loves orchids. So you can see her typing in orchid, doing a Google search. The slight sound you're hearing is her ventilation tube. She comes here, and we did not turn on accessibility mode, meaning these letters are not artificially big. These are just like you or I would use, because we wanted to push her precision of the use of that cursor. Okay, now she sees this big list, and like you or me is like, ugh, I don't want to read this big list. I just want to see some pretty pictures. So she comes back and does an image search, very, very much like we would do. And then, it says, okay, there's some pretty orchids. Let me look at those. She can G-chat, email, text, and this is the type of thing that's just starting to help people feel as though these systems are helping them. So in summary, linear dynamical systems modeling framework is quantitative and powerful, and systems neuroscience is just starting to really engage in linear dynamical system modeling. Okay, so I reviewed a lot, and I've given you a few more references, 7 here if you wish to look into them. Okay, but it's really just getting started. I think there's a long way to go, and it makes a great deal of sense why we're learning about that in class. And as my department chair, Stephen Boyd, is fond of saying, who's one of the fathers of convex optimization, LDS does unreasonably well. Even when you know a system is nonlinear, start with the linearized version of it, and it's amazing how far you'll often get. Okay, so nonlinear dynamical systems is the second point. That's also, of course, very important, but in conjunction with linear dynamical systems. We do a lot with recurrent neural networks, as undoubtedly you do as well, and then reverse engineer those learned RNNs to understand how the system is operating. And I've shown you, I've given some references here in blue, 8 through 11. And there we speak in terms of, okay, there's a fixed point here. There's a set of fixed points that create a line attractor or saddle points. This is how you get motor preparation or decision making and so forth. And the final thing is control theory. Control theory is also critical for investigating the motor system and BMI design, obviously, because when you move, you have proprioceptive and somatosensory and visual information about that movement coming back into your system, allowing you to adjust your neuroactivity in real time. And this is going to be covered in a couple of days. And synthesizing linear systems with control, bringing the two together, will also be covered, and that's a super important direction to head. So finally, the most important slide I ever show is the acknowledgements. Wonderful set of graduate students and postdocs and staff. Also our collaborators, with a special shout out to Professor Jamie Henderson, who with me directs the Neural Prosthetics Translational Lab, where we do all of our human work. And of course, especially our clinical trial participants who give very generously of their time without any expected benefit. Our alumni, who are all wonderful. Quick shout out to the arts. And finally, I'd like to thank our funders. Thank you very much.